favorite school. Go home. Please take a seat. We need to get started. Okay, let's get started. We don't want to fall behind even before the workshop starts. So it's my great pleasure to get this workshop started. So the idea is really to bring together two different fields of quantum technology and medicine and bioengineering. And the way how the workshop is organized, we have a session chair who is giving an introduction to the specific session in general, and then talks also about his or her own research, and will be chairing and making sure that we are going to stay on time with the two consecutive talks. So before we start with the individual sessions, it's my great pleasure to introduce Stephen Quake from Stanford University. He has been, as everybody knows, a pioneer in developing new technology and adapting them to biomedicine. And we would like him to give kind of an overview of how this works out, and maybe we can get inspired for quantum technology. Okay, let's move forward.
Thank you. Thanks, Peter and Amir, for inviting me. So I obviously don't work in quantum sensing, but I thought I would give a sort of idiosyncratic overview of state-of-the-art of biological measurement in certain areas, especially ones that I've worked in the past few years. And you know, what we're going to talk about, hopefully, if we get through everything, is molecular amplifiers, both analog and digital, which have been incredibly powerful for the field. Molecular counters, which have grown to be incredibly parallel and have unbelievable specificity for certain types of molecules. And then the diversity of cells in your body and how we're sort of measuring them at the frontiers right now. And finally, aspects of the proteome and how that's measured. To ensure that we're kind of starting from the same place, let me just remind you of the so-called central dogma of biology, which was articulated by Francis Crick in trying to understand information flow and protein synthesis. And he's admitted he didn't really know what the word dogma meant, because we're not supposed to be dogmatic in science, but somehow it's stuck and it's in all the textbooks. So the idea is DNA is kind of the repository of genetic information in your cells. It gets copied into RNA through a process called transcription. Then that's translated to protein by the ribosomes. And that's the way information flows to protein. It's stored in the DNA. The DNA gets copied when the cell divides and then transmitted to future generations. It can also flow backwards. It was discovered from RNA to DNA. There's something called retroviruses that do that, and they have an enzyme that'll copy RNA to DNA, so that red arrow got added later. And that's become very important technologically over time. Okay, it's a good starting spot for everybody. Yeah, great. So let's talk about amplifiers. Polymerase chain reaction was the first molecular amplifier developed by Kary Mullis in 1983. And he came up with a very creative way of doing exponential amplification of DNA using one of these enzymes that copies DNA. It had been purified, and you could use it in a test tube. And the idea was, if you start with a single DNA molecule and you have primers that match a known sequence, as well as the enzyme and the ingredients, you can separate the strands, synthesize new complementary strands, and now you've got two molecules where you started with one. And then you just repeat the process, 2 to 4, 4 to 8, and so forth. It grows exponentially. A very simple idea, but it won him a Nobel Prize because it's been so influential and powerful in biology. And there's been an enormous amount of creativity around making different kinds of amplifiers. There's ways to amplify RNA, and that's become a very powerful way to measure gene expression in cells. There's ways to amplify DNA without any prior knowledge. So in this case, you had to have a little bit of knowledge to have those primers that would bind to the template sequence, but then there are methods developed that would just amplify any DNA in a test tube, and you can amplify entire genomes that way without knowing anything about their sequence. That's been very powerful too. And there's been ways to sort of use this to amplify protein signals as well. by attaching DNA to antibodies and then going from antibody recognition to nucleic acid amplification. There's clever ways to quantitate, use this to quantitate product by looking at kind of the growth curve in an analog way that became very widely used. But then there's also ways to look at it digitally. And this idea of digital PCR as a way of molecular counting has been very powerful. And the idea is to do limiting dilution. If you took a test tube full of DNA in solution, aliquoted it, put it into many smaller test tubes, and did it in a highly dilute way, you could get on average less than 1 molecule per test tube. And then if you do PCR, you would amplify. The test tubes that had molecules would have a lot of product. The ones that didn't wouldn't. And you just count up the number of positives. That tells you how many molecules you started with. And it's independent of any nonlinearities of the amplification process. And this was first suggested quite some time ago, and it's very cumbersome to do. And it eventually got made into a commercial automated process using microfluidics. And I was involved in that in the early days. And here is an image of that. So these are multiple panels. Each one has been subdivided in little compartments with valves closing. Digital PCR was done, and the bright spots are the fluorescent products of amplification from single molecules. You just count them up. And this is now something that's very widely used. There have been a number of iterations, other commercial versions of this that let you count very high, and it's got a lot of nice properties. Another way to count molecules these days is to sequence them. And DNA sequencing has been through a revolution over the past couple of decades. And I would say it went through a major transition. In the 1980s, DNA sequencing was primarily done through a process of physical chemistry. Molecules were separated using electrophoresis and separations phenomena. And in the late 90s, early 2000s, it moved over to a more biophysical approach by using fluorescence and imaging. And today's modern DNA sequencers, which are sort of amazing machines, are at their root just a combination of an optical microscope and a dishwasher. There's automated fluidics and automated microscopes. They're just cranking through imaging many, many fields of view over the course of hours and days. And the throughput has become amazing. I'll give you a few examples. I got involved in this field in demonstrating the first single molecule sequencing, which we did back in the early 2000s. And this slide is also, I think, a good demonstration of how things can scale when it really works. Our first work, which was academic, was sort of pathetic in terms of sequencing output, even compared to the state of the art at the time. We read 4 base pairs in 30 molecules and managed to publish that. And the referees didn't like it, but we told them, just imagine how many molecules you can fit per square centimeter on a glass slide. It'll be amazing. And it sort of got through. And then we started a company to do that. And the first sort of prototype with engineering applied increased the read length to 25 base pairs and millions of templates. And that was really pretty good at the time. And then the final machine was doing billions of molecules. And it was sort of the fastest, highest throughput sequencer of its time. And the first one that was capable of doing an entire human genome on one instrument. And so this is, an example of scaling over the course of, let's call it roughly 5 years from publication. Maybe 7, 8 years to include it when we started working on it. And if you look at, this was a supplementary table we did at the time when we published my genome. I was one of the first sort of half dozen people in the world to see their genome. These were all the human genomes that have been sequenced to date. And you can see how rapidly the field was advancing as technology was innovating. The first genomes cost, billions of dollars to do if you include all the labor and equipment. But in terms of reagents, it was hundreds of millions. And you can look at the number of authors on the paper as an estimate of manpower required, number of machine runs, how many machines you need. And coverage is a technical aspect of how much you oversequence the genome. Bigger numbers are better. But as new technologies were coming online, costs going down dramatically. You can see it going down exponentially. Same thing with number of machine runs and number of authors. And it certainly didn't stop in 2009. You can take off a couple more zeros now. It's less than $1,000 to sequence an entire human genome now. And again, one instrument, one person. And it's really been an amazing revolution that went from something that required kind of a large facility to do to something that anyone could do in their lab as an individual PI, which has been just sort of a tremendous ride. And so these sequencers are basically molecular counters. You're looking at billions of molecules at a time. You're able to read out their sequence with very high specificity. And so it's like taking this digital PCR idea and scaling it up many orders of magnitude and many orders of magnitude in information content as well. Let's change gears and talk about cells for a minute. I had mentioned that there's ways to amplify RNA too. You can convert it to DNA and then sequence it, amplify it, sequence it, whatever you want. That's been a powerful way to measure sort of what we call phenotype in biology, or current state of a cell or an organism. And over time, as a community, we've managed to improve the sensitivity of these approaches so that you can now measure the RNA content of a single cell and more or less count the molecules of RNA in that cell and use that to measure sort of molecular definition of cell type. There's, maybe 250,000 RNA molecules, messenger RNA molecules in a given cell, and we can get a good fraction of them, a few percent sort of quantum efficiency, which is enough to really characterize the state of the cell. And the technologies to do that have been sort of, again, evolving very rapidly. The basic idea is you take a tissue, you use enzymes to digest it so the cells are separate in solution, and then you encapsulate them with microfluidic approaches. There's a variety of them now, where you can then amplify the RNA from each cell and barcode it, give it a DNA barcode, so you know which cell it came from. You put them all together, put them on the sequencer, and then you're able to do some sort of amazing computation. Every cell is basically represented by a 20,000 dimensional vector. That's roughly how many genes there are in the human genome. And it's been very productive. And over the last decade, I've been involved in building whole organism cell atlases, where we're trying to look at 24 tissues, try to get virtually every cell type of an organism for mouse, fly, lemur, and human. And this has been, a sort of great reference tool to understand how the genome is used in different cell types in the body. I'm not going to say a lot about it, except, here's, our current state of the art, that, the paper we're about to resubmit today or tomorrow, showing about 1 million cells across 24 tissues. Each column is a different human donor. We work with organ procurement organizations who help find organs to transplant. Whoever's not transplanted can be studied scientifically and get many tissues from the same individual, in which case we're having genome constant, age, epigenetic effects, and so forth. And we can look at things like the genes that regulate other genes. And it's been a wonderful source of insight into the biology of humans and these model organisms in ways that we couldn't do without sort of a comprehensive effort. And just, going one tissue at a time was not the way to get to the answer. I'd mentioned you can also amplify genomes, and you can do that from single cells as well. And again, the community has done that for a number of years, starting with bacteria and then going to human cells. And that's been productive for all kinds of questions, ranging from microbial diversity to understanding origins of cancer and human genetic diversity. This is from a review article on human single-cell genome sequencing. The two axes are number of cells that you're going to sequence and fraction of the genome you're going to sequence per cell, or number of base pairs. And there's obviously, as you're designing these experiments, a cost trade-off between number of cells versus amount of genome per cell. And people figured out ways to go into both dimensions. The upper right corner, still not fully solved yet. I'll throw that out there. And then finally, in the last couple of minutes, I'll say something about measuring proteins at scale. There have been some very powerful ways to do that now, using tools like antibodies and aptamers, which again started as very small-scale measurements of a handful at a time, but have now, through efforts of commercialization, been developed into highly parallel panels where you can look at 5,000 to 10,000 proteins in a blood sample. And people have been using that to look at all kinds of interesting questions, ranging from aging to disease to nutrition and so forth. Again, decades of literature on it, and real creativity in how those measurements were designed. But I will say, I don't want to give the impression that the problems have all been solved. The two major platforms to do this at scale are developed by companies called Olink and SomaLogic, and people have taken the same samples and measured it with both companies' products. And if they were making an absolute measurement, you expect pretty good correlation. The correlation is not so good. So here's from one paper, I just pulled it last night. They were looking at 871 proteins. It was a few years ago, measured in 10,000 individuals. They measured them on both platforms. And this is a histogram of the correlation coefficients between them. And they range from 0 to 1, and you can see it's a little bimodal. But there's a lot of work to do, I think, in turning this into a measurement that is more absolute and reflects the underlying reality. And there can be long discussions about specificity and sensitivity around that. The last example I want to give is returning to proteins in cells. There's been a terrific project in Sweden led by Matthias Uhlin initially, and then ultimately by Emma Lundberg, who we recently recruited to Stanford a couple of years ago, to make antibodies against all human proteins. And they, over a course of years, made something like 25,000 different antibodies, and then use them to measure all kinds of aspects of protein localization, distribution in cells and tissues. And their website is a really fun one to explore and very highly visited. Just sort of another example of how to do protein measurements at scale, not in blood, but in cells this time. And they've got an amazing database of images that are really fun and interesting to explore. And with that whirlwind tour, I'll thank you and hope I've set the stage for some really fun discussions. I'm looking forward to hearing all the talks today. Thanks.
We guess we have maybe time for one question, two questions. Yep, back there.
So for the genetic transcription, what is the error rate for the first part that you talked about?
So you got a question of error rates in sort of copying molecules, right? And when just sort of 1 in a million when a genome is being copied for cells to divide. For transcription, making RNA from genes is much higher. It's of order, I think, a part per thousand, something like that. And the cells have to figure out how to work around that. Then there's errors in the translation as well. And these questions of how you make faithful molecules has been an interesting one. And John Hopfield very famously had this idea of kinetic proofreading around protein synthesis translation, and it wasn't quite right, but it's provoked a lot of interesting thought on how that's done. So it varies depending on which molecule is being copied, is the short answer. DNA, most faithful, protein, least faithful, because you can tolerate the errors more.
Great, maybe time for one more question. Randy?
Yeah, I guess comment about, this provocative comment about the differences between Olink and Semologic. Can you talk a bit more about kind of maybe hypotheses as to why those two data sets diverge so drastically?
Yeah, I mean, so full disclosure, I was on the Semologic board for a couple of years, and I kept telling them, you've got to do this head-to-head and understand the limits of your measurement. And they were more interested in getting more sales. And so I never managed to carry the day of, having them get the most rigorous possible measurements. And that dynamic just sort of exists in companies because of the commercial pressures of being there. So you need a few independent academics who are willing to do the head-to-head measurements. I think by looking at the independent measurements in the literature, I think most people would agree that Semologic has a bigger panel, more proteins, so it's better in that respect, but they have less specificity. So there's a little more cross-reactivity of the reagents. And that goes to the sort of intrinsic design. They've got a single aptamer, whereas, so it's only binding one thing, whereas Olink has got 2 antibodies, so you get the extra bit of specificity there. So Olink has fewer proteins in the panel, but probably better specificity.
Thank you.
All right.
Thank you very much.